We've completed our discussions talking about methods of phylogenetic reconstruction, and we still have a few wrap-up things to do in the area of general phylogeny, and one of those is supertrees. So supertrees really are a response to larger and larger data sets, and some of the issues that we talked about when we looked at our different methods about how long analyses of data sets can take. And so realize that in many cases, thorough analyses of these big data sets are quite prohibitive. They're time consuming, they take lots of computational resources, and so it can be a problem. And so one response to that issue of, well, I just don't have enough time or enough resources to do a good analysis was a supertree approach. And a supertree is really a approach that allows us to take different analyses and somehow put them together. So it is not an analysis of primary data, it's an analysis of analyses, or what we might call a meta-analysis. So it does not rely on the primary data itself. So remember we talked about when we were looking at distance methods, there was a little bit of an issue with, well, we take our primary data in the form of DNA sequence and we turn it into distances, and then we use those distances to calculate a tree. Well, this is maybe even worse because we do whatever methods we want first, maybe even a variety of different methods in different analyses. And then we do a second round of analyses based on the results of the first. And so the idea from a statistical or scientific point of view is that every time you do something, there's a degree of uncertainty that's introduced. And so if you have a degree of uncertainty in your trees, and then you're analyzing those and doing a secondary analysis to figure out a super tree, right? Then you're adding on another layer of uncertainty. And so although it might be useful for these large data sets and maybe even help us to get the overall phylogeny of all living organisms, or what we call the tree of life. And there's a website, if you want to look it up, it's TOLWeb, T-O-L web, I think .org. It's kind of a cool site. You can look at it. It doesn't it's conservative. It doesn't update as frequently as maybe the scientific community reaches consensus, but it gives you a good overview of all living creatures. And so really there's some super tree work there and some things put together to try and figure out the entire phylogeny for all living organisms. And this is fundamental because really we could, in theory, although it's very difficult in practice, figure out relationships because of this uniting idea in evolution that all living organisms share common ancestry. Now, a super tree, there are a couple of different ways to do it. The first we're going to talk about is an informal, but all of these methods allow us to put a variety of different analyses together into a single image or a single diagram representing relationships. So first, let's talk about informal supertrees. Okay, an informal supertree, as it sounds, we don't do any objective analysis. There's no strict analytical way to do it. So for instance, we might do a phylogeny of amphibians using whatever genes we think are good and whatever taxon sampling we can. We do an amphibian of reptiles, maybe different genes or maybe some of the same ones. We do a phylogeny of mammals, and we do all those separately, and then we stick them together in a way that maybe other analyses, maybe even a fourth analysis, show those deep relationships. So we'd say, oh, here's our mammal tree. We just kind of graft it onto what we think the phylogeny is. And we end up with an informal supertree. And so we have three different analyses all represented in one tree. Now, of course, depending on results of these different sub-ones or our understanding of the deeper relationships, different people might have different informal supertrees. And so there's really no objective way to do it, particularly if there's any disagreement between the underlying phylogenies, right? So if there's conflict in those subtrees, an informal supertree just really does not work well. So people wanted to come up with more formal ways to analyze our previous phylogenies and try and make a supertree diagram. Okay, so formal analyses are when we do some sort of a step-by-step algorithm, if you will, or heuristic rubric, right? A rubric is kind of, if you do this, 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 and this, you'll get a good grade on this assignment, but you have rubrics for other things. But so basically, we have a set way to do things to make a formal supertree, and that's why we call them formal. The first type we're going to talk about is an agreement supertree. And basically what an agreement supertree does, and you can do this through a formal matrix analysis. There's several methods to do this, and we're not going to get into the methods. There's a table in the Beninda Edmonds paper. If you want to look at it, it goes over and reviews some of these, but don't spend time memorizing them unless that's your jam. It won't be on an exam, okay? But just know what an agreement supertree is. And so we do some sort of analysis by making a matrix or doing some sort of step-by-step category, and then we represent all of the species with an overview. And notice that we didn't just simply prune because, for instance, some of the green ones are in different places. So we only have species I represented in this one analysis. And it's most closely related to E and H. And since E and H occur here with F and G put inside, notice that some species are represented in some analyses and not in others. And so we don't have complete coverage. And so as long as there's no conflict between these trees, you can go through a number of steps. You'll probably get the same formal supertree by looking at agreement. And if they're complete agreement, no matter what method you do, you'll get the same result. But sometimes it's not 100% agreement. And if you have 5 or 6 different trees that go into it, that's not too bad, because then maybe you could say, well, if 3 of the 5 trees show this set of relationships, that's what I'll put. And if there's conflict in the minority, we won't include those. So there are ways to get around this, particularly if you have a lot of trees going into the analysis. But inevitably, you're probably going to have some disagreement. And so the methods are just formal ways to figure out how we deal with the areas where there is disagreement. Okay? Now, a separate way, a different way to do a formal supertree is called an optimization supertree. And in an optimization supertree, we would maybe make a matrix. We've got this diagrammed here. This is, let's move on to the next one for optimization supertrees. One way that you can do that is you can reconstruct a brand new matrix. But the matrix is not your primary data. The matrix is simply, is a group represented in my analysis? So in tree number 1, we have B, C, and D all together. And so we'd say, oh, look, B, C, and D all together, and A is outside of that. Right? And so we're just basically making a very simple matrix. And for node number 2, we say C and D are together, and A and B are different. And so we just do this, and there may be disagreement. So this is a way to formally incorporate disagreement into the matrix. And then you could do like a parsimony analysis. You can't do really maximum likelihood on these. So you'd probably do a parsimony analysis on your new matrix and figure out what the phylogeny was. And so it's a good way to do it, but again, if there's conflict, you're going to maybe have some issues. You might end up without as much support as you originally had, but you make a matrix. So matrix representation is one way to do an optimization supertrees. There are other ways. Again, you can look at some of those different methods in the book. We're not going to go any deeper than that. Okay, so there are some major criticisms and problems with supertrees. And in fact, many people will not even use supertrees because of these criticisms. So let's deal with them. One we already talked about, right? We're one more step removed from the primary data. It's an analysis of analysis, which adds extra probabilities of uncertainty and error into our final results. And so many people don't like it for that reason. Number 2, we might lose what has been termed signal enhancement when we combine the final outcome. So for instance, let's say that in a certain data set you have a signal that's telling you about some relationships, but it's kind of swamped out by the noise, like the random mutations, the back mutations, the things that make doing phylogenies complex. If we combine data sets and then do a single analysis on just the overall combined data set, which is the more formal, the more standard way to do it, then all of those signals, those phylogenetic signals that are kind of swamped out in the different analyses are going to get together and then maybe come out, and all the background noise, if you will, the background confusing data would be swamped out by the enhancement from the multiple signals. But if you just have kind of a little bit of a mess in one phylogeny, a little bit of a mess in another one, and you don't put the primary data together, you don't get that signal enhancement. Number 3, novel clades are not supported in the source data. So sometimes, especially when you're doing those optimization, those formal optimization analyses, you end up with a clade that's not in any of your previous phylogenies. It's novel, it's brand new. So how do we explain, oh, look, here's a set of relationships that's not in any of them? And that's possible. It doesn't happen a lot, but it is possible, and so that's a problem with the approach. This is bothering me. I don't know why there shouldn't be 2 3s, but 4. And lastly, If we're not careful, then we might inadvertently replicate our source data. So for instance, if we're doing a agreement supertree and 3 out of those 4 trees that we're using all had 18S genes in it, then basically we're representing that 18S data multiple times in our final analysis. And so that can be really tricky. And so if the 18S is biased in some ways, some of that bias might be magnified if we have it represented in multiple original analyses. And so supertrees are not really considered the best way to do it by most people, but sometimes they're a good alternative, and they do add a useful point of view and maybe another useful way to look at and get into relationships of organisms. So with that in mind, despite these criticisms, sometimes people are now designing approaches before they actually have the data. So all of these methods we talked about are mostly what we would call an ad hoc, or maybe more precisely, a posteriori method of figuring out relationships based on those previous analyses. So what I'd like to do is just very quickly talk about what a posteriori means. It's a Latin phrase, and a priori mean. These are pretty obvious and easy for these two. Ad hoc is kind of a modified version of a posteriori. So let's just be clear about definitions. A priori means before the fact, or before you do something. So you come up with a design or an approach before you actually do something. A posteriori, as the name, many of our English words and in other languages also are Latin-based, so this is helpful. A posteriori means after the fact. Or in other words, you've done something and then you go back in and try to fix it, or try to do, in this case, a supertree analysis based on what's already been done. And of course, ad hoc just simply means as is or like it is. And so an ad hoc committee means for this specific purpose. It means it's put together for one specific thing. So you can kind of think of these a posteriori also as ad hoc, because they're done just for that one specific result of trying to find a supertree. But now people are coming up with designs and approaches before, rather than just going through all the data and analyses that are already available. They say, hey, let's do this in a little bit more of a defensible manner. Okay, so one way to do that is discovering method. So we get our data set, and maybe we do a very quick phylogeny like a neighbor joining tree. We get a very basic, maybe rudimentary, but decent overview of relationships. And then we subdivide that with overlap so that there's going to be some areas that they share and then some that they don't. We subdivide that into subcategories, and we analyze those trees and then use one of our methods, usually a formal, either an agreement or an optimization method, and get our supertree. And so if we design these ahead of time, then we're not going to have only like 1 or 2 branches that are shared between them. We have a good amount of sharing, but it's kind of a divide and conquer approach. So that's the discovering method. The other method deals with the primary data. And this was originally, it's not as important anymore, but still plays a major role in supertree methods, because we have these big data sets, but we're missing data for some of the species. So we have a species here, and we have genes along this side. So we have gene 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different genes. Some of them are represented in some analyses, others in other analyses. And so we don't have all of the taxa represented for every gene. And so then what we would do is we would kind of look over and say, are there any what we call a biclick, which is a group of species where we have a large, or maybe even complete, in this case, overlap of represented genes. And if so, then we take that as one data set and we analyze it. Oh, look, here's another data set where we have 3 species with every single gene represented. 1, 2, 3, 4 species with 3 different genes represented for all 4 of those species. And so that's how we would divide and conquer. In this case, we're doing it based on what gene samples we have, rather than just on any sort of starting estimate of relationships. So be familiar with both the biclick method and the discovering method. Know that they are a priori methods for supertree approaches, and that's the level of detail and specificity that you need to know for supertrees.